for allowing me to take this tour. I want to thank Tim right away, who's been tremendously helpful in setting up all the AV and solving the challenges that my systems seem to bring here, American systems seem to bring here in the UK. A little bit about me. Obviously, I'm from the United States, not from here. I grew up in Wisconsin. I grew up in a Christian home, a creationist household. My dad was a dentist. My mother was a nurse. I was homeschooled through 8th grade. I grew up under a creationist curriculum. When I went to high school, so grades 9 through 12 in the United States, I went to a creationist high school. There were 3 Christian options in the area. My parents chose one that was explicitly young age, young earth creationism. I went to the University of Wisconsin Parkside for my undergraduate degree, my first degree, Bachelor's of Science in Molecular Biology and Bioinformatics, a mouthful just meaning life at the smallest levels, and then using computers to analyze life at the smallest levels. Did my PhD at Harvard, wanting to study cancer. Harvard has the Dana-Farber Cancer Institute. I remember the first year I came, the summer before I began, fall of 2003, I visited Boston and interviewed with one of the cancer researchers there. He said, "Glad to hear you wanted to work on cancer. There's 15,000 people here working on cancer." So it was a little intimidating, but it was a thrilling ride. I worked on adult blood stem cells at that time in 2003, 2004. This connection between the behavior of cancer and how our cells normally behave, how our tissues renew themselves, there was a growing connection. So my thought was, if we can understand how the body normally renews itself and cells divide and that sort of thing, perhaps we can understand how things go awry in cancer. I graduated in 2009. I spent 6 years at the Institute for Creation Research that was founded by Henry Morris. He'll come up in my talk in a few minutes. In California, it's currently in Dallas, Texas. I was in the research department there 6 years developing a biology research program, published a number of papers. I've been 3 years at Answers in Genesis in Kentucky. So this is the organization that has the Creation Museum. We're just outside Cincinnati. We also built a life-size replica of Noah's Ark. So if you've ever come to the United States in the area of Cincinnati, I highly recommend those 2 attractions. I have a wife and 4 kids. They're back home. Thankful for technology, FaceTime, Skype, so I can see them. The kids are ages 5, 4, 2, and 3 months. So life is never dull, and they're a lot of fun. What I want to do tonight, I've written a book called Replacing Darwin, The New Origin of Species. You can't replace Darwin in 60 minutes, so I'm not going to try. That's why I wrote a book, Replacing Darwin, with extensive footnotes to try to do that. Instead, what I want to do is give, in the few minutes we have together, 5 reasons to reconsider Darwin, to revisit Darwin. And if you walk out of here and say, this is a different debate than what it used to be, I think I've been successful. Now, I wrote my book for an audience assuming that they don't agree with me, and I'm sort of going to deliver this lecture that way. So even if you aren't in agreement with me, this is just the approach I'm taking. And I want to walk through 5 scientific reasons why I think it's time to revisit what's been the consensus for a century and a half. First reason I think there is to reconsider what Darwin wrote is that Darwin took a massive scientific risk, and I'll explain this in a moment, but I don't think this is a terribly controversial point. So Darwin's book is titled On the Origin of Species. What's a species? Zebras are a type of species. There's actually 3 recognized zebra species. We recognize them by their stripes. We define them almost by their stripes. We define them sometimes by their genetics, sometimes by reproduction. We define species by their traits. Well, one odd-looking individual does not a species make. You have to have a whole population. And there's 2 ways you can get a population of visually similar, genetically similar individuals, and that's to have the ancestors that live forever. No one believes that. Or you have a set of ancestors that reproduce and pass on the information for those traits and get a population of similar individuals that way. So the fact that you have this inheritance process means that at the end of the day, this question of the origin of species is a question of the origin of the traits that define those species. And since traits are defined by inheritance, this is ultimately a genetic question. The origin of species is ultimately a genetic question. And if for no other reason, it's a genetic question because Darwin proposed a fundamentally genetic hypothesis. At the end of the day, one of his main theses is that all of life exists on this large interconnected family tree. Humans are just one branch of this tree that includes primates and reptiles and amphibians and elephants and everything that lives on this planet. Well, the only direct scientific record of a creature's genealogy is genetics. So this is ultimately a genetic question we're trying to evaluate. What's interesting, though, is if you look at the history of genetics, it doesn't begin until a few years after Darwin publishes his book. Darwin publishes the first edition in 1859. Gregor Mendel, the father of modern genetics, publishes his tedious but comprehensive work Crossing the Common Garden Pea Plant. He publishes that work in 1865, 1866. Mendel gives us some profound principles of inheritance. He says nothing about DNA. Ironically, Mendel's results were lost from the scientific community for several decades, rediscovered around the turn of the century as scientists began to study the inner structures of sperm and egg and how those intercellular structures behave from generation to generation. And there was a light bulb moment as they connected the behavior of chromosomes, which I'll tell you more about in a minute, to Mendel's principles. And so they said, okay, chromosomes must be the substance of heredity. Here's a diagrammatic version of a chromosome. What dominated scientific debate for the next several decades was chemically which part of chromosomes encode heredity. Chromosomes are made up of two major molecules, obviously DNA here, that twisted ladder structure, and in yellow, though, those plugs are proteins. So there was a big debate as to whether it was proteins or DNA chemically that encoded heredity. And proteins dominated the debate for many years. It took a lot to overturn that. And to make a long story short, that was one of the most instrumental discoveries in overturning that view and shifting scientific opinion towards DNA as the substance of heredity were the discoveries of Watson and Crick and colleagues, famous, of course, for giving us the double helical structure of DNA that we all recognize now as iconic. One of the main things I want you to notice, though, in this history is the date. So it's this discovery, this structure, that settles the debate over DNA or protein. And that settling of the debate happens in 1953, nearly 100 years after Darwin wrote his book. This settles one question, as every good scientific research project does. It raises new ones, and the new question that was raised is, what are the DNA sequences of the various species around the globe? And this graph depicts how our knowledge of the DNA sequences of species all over this planet has grown dramatically only in the last few decades. So the first DNA sequences were discovered around the late 60s. Our own DNA sequence of our species, the human DNA sequence, was discovered 2001, chimpanzee 2005, and there's been an explosion only recently. So if the origin of species is fundamentally a genetic question, we really haven't had the direct tools to investigate this question until recently. So in that sense, Darwin took a scientific risk, and again, I don't think this is controversial. Francesco Ayala said Darwin knew 1% of what we know today. He said he knew the most important 1%, but he knew a fraction of our knowledge today, and so in that sense, he's taking a risk, trying to answer a genetic question without any genetic tools at his disposal. That's one reason I think it's time to stop and say, let's give this a second look. Second reason to reconsider Darwin, reasons to revisit Darwin, is that creation science has changed. It's a different debate than what it was. There's an element in which it hasn't changed. There's a way, though, it has. So let me back up to the beginning of this debate. I'm going to start this debate in 1758. That's the 10th edition of Carl Linnaeus's book, catalog Systema Naturae. He's a Swedish biologist considered by evolutionists and creationists alike to be the father of our modern classification system. And like many of his contemporaries, Linnaeus believed that the species he was documenting, cataloging, were fixed units of God. Now, what I forgot to mention here is I'm starting this debate here because Linnaeus gives us our term species as we use it today, and you really can't debate the term species until you agree on the definition. So I'm starting this origin of species debate with him. So back to his views on the origin of these species. Like many of his contemporaries, he thought that examples like this creature, the Arctic fox, were directly created by God, put in their current locations, not the result of evolution. He was a creationist. Now, why would he think that? One of his contemporaries was William Paley. He gave us the watchmaker analogy, that if you find a watch on the ground, you immediately think it's designed, not the product of time and chance, because you can see the parts matched for an obvious purpose. And that sort of thinking was applied to biology. You can see the parts of this fox, its white fur, its thick coat, seem matched to the snowy, harsh Arctic environment in which it finds itself. It blends in, at least to our eyes, with its white coat. Thick fur helps it survive in this harsh conditions. You naturally think it's designed for the Arctic. Linnaeus knew of other fox species like this red fox that exists in Europe and Asia. It does not have a stark white coat. It has this reddish coat, and you can see its environment is not always stark white, but it has multiple colors. There's actually 2 foxes in this diagram. Not the reflection here, but this one here, and there's one hidden in the bushes here. So even again, to our eyes, separate environment, different parts of the fox, different colors, I should say, yet they seem matched to the environment in which it exists. So this is one of many examples we could cite that drove the thinking of scientists in that day. And the thought was God designed these creatures for their environment. And this leads to 2 conclusions on the origin of species. Number 1, they were designed for the environments in which they currently exist and placed there from the start. You wouldn't naturally think that God put that Arctic fox in the Sahara and then said, go find your way to the Arctic. Doesn't make sense. So God puts the Arctic fox in the Arctic from the start. He puts the red fox in Eurasia, Europe and Asia from the start. And so these 2 species, number 1, don't have a common ancestor. They're created separately. And number 2, they haven't migrated. They were just put there in that current location. That's the view that Darwin was trying to overturn in 1859, and he was very effective at refuting this view of the fixity of species. What many people don't know, though, is that Linnaeus' view that I've just depicted here represented the views of his early career. Later in his career, he changed his view. He worked primarily with plants, and what he discovered was that some of his plant species could actually intermingle. So this raised new questions in his mind. He was thinking, okay, if God creates species, kinds, separate, unchanging, fixed laws of creation, but some of them can change by intermixing, this made him rethink. So as a modern example in animals, horses are one species, donkeys are another species. They can be crossed to form mules. We have at the Creation Museum, our petting zoo, zorses and zonkies. So horses, donkeys, zebra, all separate species. Well, horses and zebras can interbreed, form that zorse there on the left. Donkeys and zebras can interbreed, form that zonkey there on the right. These sorts of phenomena led to the following view later in his career. Linnaeus stated that God created as many individuals as there were orders, that God later mixed these to form genera. So order is a higher level of classification. Genera is the plural of genus. It's the level of classification above species. So he's not talking about God creating species anymore. He's thinking in very different categories. And then Linnaeus says nature in turn mixed these genera to form species, and fate mixed these species to beget varieties. And as a specific example, Linnaeus proposed that dog, wolf, and fox, all separate species, belong to the same created genus, but they were products of hybridization. So let's generalize what Linnaeus was saying. So if he says that nature can mix genera, the Arctic fox and the red fox not only are separate species, they're each in their own genus. So if Linnaeus says that these genera, one genus can mix with another, well then surely the units that God created are not species, not even genus, but probably more appropriately at the level of family. So the dog-like family, technical term is Canidae, includes creatures as diverse as foxes and wolves and coyotes and jackals. So this is a very different view than the fixity of species. Darwin was not aware when he wrote his work that Linnaeus had changed his view. He didn't find out till later in his career. Well, Linnaeus' view is the one that Henry Morris, and I'll tell you more about him in a moment, picked up. He's considered, at least in the United States, the father of the modern young earth creationist movement, young age creationist movement. He was a professor of hydrology, studied water, PhD at Virginia Tech University, chair of the engineering department. He wrote a book early in his career, That You Might Believe. He was a Christian, so he had many Christian students coming and asking him questions about science and the Bible, and he wrote a book, That You Might Believe, and he said in that book as early as 1946, it's well to observe at this point that the Bible does not teach the fixity of species. Thus, it is probable that the original Genesis kind is closely akin to what the modern systematist, modern classification scientist, calls a family. And since Linnaeus' day, since Morris's day, we have more and more and more examples of different species being able to cross. This is a liger, a cross between a lion and a tiger. Lions and tigers are separate species. And the result of all this crossing has resulted in the same general rule. So modern creationists still hold to this level of family as a good approximation of what that created kind is. Now, if you take this logic one step further, okay, family, not species, but you just said that dogs, wolves, coyotes, foxes are part of the same family. In other words, what I just said is there are many species within a family. That implies then that the modern creationist view is okay with new species forming. And in fact, that is the modern creationist view. God creates families, ancestors that sort of represent this family level, and within these families then new species can form. Modern creationists would say that one family cannot change into another as a general rule, but within the family all sorts of variety is possible. So in these next few slides I'm going to illustrate then some of the variety that young earth creationists would say has arisen from those ancestors: foxes, jackals, dogs, all sorts of species, 30 plus species, and 300 plus breeds of dogs, all from a common set of ancestors, common pair. Using the zebra, horse, donkey example, as I mentioned, there's 3 recognized living species of zebras. Those, along with the 1 wild species of horse, they're on the left, and then in the center and on the right, the 3 wild species of asses, onager, kiang, and African wild ass, plus all the breeds of horses and donkeys. Those 7 species and the breeds all go back to a common pair, is what the modern young earth creationist view says. In some cases, for example, in birds, the young earth view has gone to perhaps modern standards to a wild extent, something that would seem wild in the context of 1859. So I'm going to give you 2 slides of some of the variety within what modern creationists would call the sparrow-finch kind. And you can look just at the plumage here in these birds, very diverse. Based on breeding, the number of species that young earth creationists would say formed within this group is over 1,000. It's a lot of new species by a creationist point of view. And they say these came from ancestors on board Noah's Ark, whether it's mammals or birds. And as a general rule of thumb, young earth creationists would say, if we were to generalize those examples I just gave you, that God commanded Noah not to take every species on board the ark, not even representatives of every type of creature. There's no need to take aquatic creatures on board because it's a flood. There's unlikely that Noah brings insects and invertebrates. Generally, the modern earth creationist view says that Noah's commanded to take mammals, amphibians, reptiles, and birds. And today, there's over 30,000 living species of mammals, reptiles, amphibians, and birds, and less than 2,000 families. And even if you include all the extinct species in those categories, you're under 100,000, but they come from just a handful of ancestors on board the ark. And young earth creationists would put that flood event about 4,500 years ago. So think about that. Modern young earth creationists say that there's over 30,000 new species that form in a few thousand years. Some of my evolutionary colleagues have recognized this shift, and they've accused younger age creationists of being hyper-evolutionists, because we believe in so many species in such a compressed time scale. But my point here is that creation science has changed in 2 respects. First of all, in its position on speciation, going from a species fixity view, that's the view that Darwin tried to overturn, to a third position. It's not evolution, it's not species fixity, it's sort of a intermediate between the 2. Transitional form, in a sense. There's a second way creation science has changed, and I want to put this on a timeline so we get some of this history, so you can see how this practice of science has evolved over time within a creationist framework. So as I mentioned, I'm going to start this species fixity view with Carol Linnaeus and William Paley. That lasts for about 100 years until Darwin overturns this. And if you look at the creationist reactions to Darwin, they take a particular tactic. So here's Samuel Wilberforce, Bishop of Oxford. We were in Oxford a few days ago. I got to see the monument erected outside the Oxford Museum of Natural History, commemorating the famous exchange between Wilberforce and Thomas Huxley at that location in 1860. Wilberforce formally wrote up a review, a scientific critique of Darwin's work, and this is a quote from that. I want you to notice the way in which he reasons about Darwin's work and what he says against it. So he says, if it were possible to proclaim more distinctly by one provision than another that the difference between various species was a law of creation, and not, as the transmutationists, evolutionists, maintain an ever-varying accident, it would surely be by the interposing such a bar to change It would surely be by the interposing such a bar to change as that which now exists in the universal fruitlessness, which is the result of all known mixtures of animals specifically distinct. That was a long Victorian sentence, and for Americans like me who have a smaller vocabulary than British English speakers, I'll summarize it for you, summarize it for myself. What Wilberforce is basically saying is, look, species are basically defined by reproductive isolation. The fact that you often can't cross species, the fact that even, for example, the product of a horse and a donkey crossing and producing a sterile mule, if that doesn't echo and give you the stamp of God separating species at creation, what does? So that's the view in 1860. And it's largely an anti-evolutionary view, and his argument is basically evolution can't happen. How are you going to cross that barrier to new species forming and to species mixing? Evolution wins the day, and that's been the rule in the professional scientific community ever since then. In terms of professional scientific opposition to evolution, there really isn't much, if you look at history, in the way of a national movement, at least in the United States, for nearly 100 years. It gets restarted by a second book that Henry Morris publishes while he's still at Virginia Tech, chair of the engineering department. He collaborates with John Whitcomb, a theologian who had done work at Princeton. They wrote a book, The Genesis Flood, that was enthusiastically received by the Christian community. I was born in 1980, but those generations prior to me have told me that for many Christians, this answered so many of their questions. How do we put science and the Bible together? They didn't have many answers. This book comes along and, aha, it makes sense for them. Morris quickly draws a large following in the United States, so that within 2 years, there are enough professional scientists who are in agreement with him, they form a professional society. The Creation Research Society is founded in 1963. This society eventually publishes a textbook for use in the school system. Perhaps not surprisingly, by 1969, life becomes difficult for Morris at Virginia Tech because he's written a book that contradicts so much of what his colleague says. He takes a sabbatical, decides to leave Virginia Tech, and founds in 1970 the Institute for Creation Research, where I was on staff for 6 years prior to coming to Answers in Genesis. Duane Gish also joins Henry Morris on staff. Duane Gish is a PhD biochemist, and during the 1970s in the United States, they do about 100 debates, many of them on university campuses. And the evolutionists, in writing about this time, concede that they were very unprepared for this. They say they admit they did a terrible job. Morris and Gish debated the leading evolutionists of the day. Now, what was so surprising about Morris and Gish's strategy that caught the evolutionary community off guard? This is what Morris says about their strategy during debates at that time. So there were thousands of students coming out. The students were writing this up in a positive way in the student newspapers. There was a real wave of interest in creation science. So Morris says this, and gives his 4-part strategy for debating evolution in these public venues. He says, we are always careful to stick strictly to scientific arguments, especially using the fossil record to show that macroevolution has not occurred in the past. That's the first point. He says, gaps in the fossil record, no evolution in the past. Number 2, the characteristics of mutation and natural selection to show that it is not occurring in the present. So basically, they would point out, it seems that mutations are largely negative. This points towards degeneration, not evolution. So they'd argue, number 2, evolution does not occur in the present. Number 3, Morris was famous for this, the laws of thermodynamics, to show that it could not occur at all. So he would say that the second law of thermodynamics says the universe is going towards disorder. Evolution seems to require a movement towards order. How does this make any sense? And Gish was famous for saying and talking about the origin of the first life forms within an evolutionary model and arguing that complex functioning systems could not arise by chance. Probability rules this out. That was the 4-part, largely anti-evolutionary strategy, similar to the Wilberforce strategy. That was the tactics they employed in the 1970s. The results of this, again, besides just general enthusiasm for the creation science position, were legislation in the United States. Many states began to then adopt laws that said you need to teach creation science in the classrooms. And the results of this building enthusiasm caught the attention of the evolutionary community in the United States, so that by 1981, the National Center for Science Education was founded. This was founded for the explicit purpose of opposing the teaching of creation science in U.S. public schools. Eugenie Scott, shown here, was one of the more famous past presidents. 1982 was a watershed year in the United States. the state of Arkansas had a law on the books mandating the teaching of creation science. That was challenged, brought to a federal court trial, and the judge overturned it. I say this is a watershed year because that trial got national attention, sort of marked a point in time at which interest had grown and had caught the attention of the evolutionary community, and from that time forward you see a long string of anti-creationist material. This has set the context then for our modern debate. This is the context I grew up in. I was born in 1980, and as an anecdote, I'm going to make some points here in a few slides that I thought were normative for the past 150 years. In fact, they're recent, and I didn't realize this because of the year I was born. If you look for anti-creationist literature in the 1970s and the years prior, you really don't see much. And so I was trying to document a particular quote, a particular strategy on the part of the evolutionists from the 1950s, went and found in our library textbooks from the 1950s, and I couldn't find any. Apparently there wasn't enough creationist opposition for the evolutionists to even think it was worthy of a mention. That changes by the 1980s, and you see a long list, a long string of anti-creationist literature, and I want to show you some quotes from these, because there's a common thread now in how evolutionists deal with creation science, different from being caught off guard in the 1970s. Niles Eldredge, one of the famous scientists of the time, famous for proposing punctuated equilibrium along with Stephen Jay Gould. He wrote a book, The Monkey Business, A Scientist Looks at Creationism. This is a 1982 book. And he said this about creation science. He said, creation science isn't science at all, nor have creation scientists managed to come up with even a single intellectually compelling, scientifically testable statement about the natural world. At least 95% of all their reams of privately published books and pamphlets are devoted to an attack on conventional science. So he recognizes the Morris and Gish strategy of evolution can't do this, and he says that's just an attack, that's a negative strategy. The problem with creation is they pose no testable hypotheses, make no predictions based on their views, no observations worthy of the name. So I'm going to show you a few more quotes here, and I want you to see a common thread. If you look at the text of the court decision itself, this is what the judge said. The essential characteristics of science are, and he lists 5 of them. Number 5 is that science is characterized by being falsifiable. And the judge says creation science fails to meet these essential characteristics. It is not testable, is not falsifiable, and then therefore doesn't belong in the science classroom. You look at modern evolutionary textbooks. I have this in my office back in Kentucky, in the United States, the 4th edition of Douglas Fudiyama's textbook on evolution, typically used in the United States for university students in their 3rd year, for biology majors, biology students. He has a whole chapter now dedicated to dealing with creation science. And he makes this point. He says the most important feature of scientific hypotheses is that they are testable, and then says creation science isn't science because it's not testable. I've listened to 2 debates this year between evolutionists evolutionary proponents and a creationist. The gentleman on the left is Herman Mayes. He's an evolutionary biology professor at Marshall University in West Virginia in the United States. Aaron Ra is a more famous atheist there on the right, and both of them basically gave the same challenge to their creationist opponent: give us a testable, falsifiable prediction. And what all these quotes have in common, what I want you to see, is a view of the nature of science that goes back to a man named Karl Popper. Popper lived and died after Darwin wrote his work, and Popper gave us this element of science, this idea of falsification. Popper's falsification methodology holds that scientific theories are characterized by entailing predictions that future observations might reveal to be false. So in a nutshell, the way science works is you observe, you infer sort of a general rule that you think might be true, and then you go and test it. And you test it with experimentation, but the purpose of your experiments are not, and this is very counterintuitive, not to prove your rule to be true, you try to disprove it. And if your rule that you think is true, and you try to disprove it, and you can't disprove it, and you try again, you can't disprove it, and you try again, you can't disprove it, if your idea survives that over and over again, then you begin to think, I bet you this is true. That's sort of the counterintuitive way of doing science, falsification. The point here is, this is the standard to which creationists have been held for the past 40 years. Give us a testable prediction, is what I'm hearing from evolutionary opponents. Last fall, October, I published a book, Replacing Darwin, in which I laid out testable predictions to meet this standard. So I'll give you a specific example, drawn from some of the data I discussed in Chapter 6, applied now to birds. So there's 11,000 living bird species today. For sake of argument, let's assume within a creationist framework that they arose from a few hundred ancestors on board the ark, and at a constant rate, just to keep the math simple. I'm going to ignore those few hundred in my math and just use 11,000 because it's a nice round number. And I said earlier, divided by 4,500 years gives you an average rate of species formation of 2.4 new bird species per year. It could have been that there was a burst of speciation immediately following the flood that then tapered off to the present day. You still get an average rate, just as sort of a ballpark number, 2.4 new bird species per year. Very different then from what the evolutionary model might propose. So the modern evolutionary model says birds evolved from dinosaurs. So to get from dinosaurs to birds, you have to go through transitional forms. Dinosaurs go extinct within the evolutionary framework about 65 million years ago, but given that lag time to form modern birds, and if you look at the evolutionary literature, they typically put the origin of modern birds beginning around 35 million years ago. So let's use that number. 35 million years divided by 11,000 species gives you an average rate of one new species every 3,200 years, much slower than what I just told you. And to give you a side-by-side comparison, within 3,200 years, at the rate of species formation that I just proposed, 2.4 per year, in 3,200 years you get nearly 8,000, 7,680 new species. So there's orders of magnitude difference, thousand-fold difference between these two models. Not surprising, because we're dealing with timescales, 6,000 years, millions of years, that are orders of magnitude different as well. But the point here is this number is something you can test and try to falsify. You can do experiments on this. Now, some of my opponents have said, well, we've already done the experiments. No one's observed the formation of a new species within the last few hundred years, as long as we've been studying species. And I said, well, let's hold on a minute. When does the modern discipline of classifying life begin? Well, it begins with Carl Linnaeus. He lives in the 1700s, so roughly 300 years ago. And we have, if you look at documentation for the modern bird species, and they give the author, who's the author responsible for discovering the species, you won't get things older than Linnaeus. So the bottom line is we've discovered 11,000 new species within 300 years. We've been discovering, on average, 37 new bird species per year, much higher than the predicted rate at which species form. So really the question that we need to ask is, how many of these bird species discoveries represent, number one, either the discovery of a species that has always been in existence, but we just didn't pay attention until now, or versus number two, how many of these 11,000 species documented in the last 300 years represent bona fide new species formation? Well, how would you do that practically? Part of the practical challenge in testing this hypothesis, in evaluating that rate of 2.4 new bird species per year, is that there's anywhere from 9 to 30 different definitions of what a species is. And to give you a sense for why there's variety, some species have obvious visible differences. If you look closely at these zebras, it might be hard to see at the back, but the plain zebra has stripes that cross the belly. It differs from the mountain zebra and the imperial zebra, which their stripes do not cross the belly. You can see in the plain zebra, it's got fairly thick stripes. The mountain zebra and imperial zebra have fairly dense stripes. The point is there's obvious visual clues we can look to say this is a species that's different from this other species. In some cases, though, for example, the onager and the kiang, I, for the life of me, can hardly tell the difference. They're so visibly similar. But if you look at their cells and their genetics, they have different chromosome numbers. They have obvious and profound genetic differences, so they're labeled different species. Classically, species have been defined by their ability to reproduce or to not reproduce. It goes back to Ernst Mayr in, I think, the 1930s. The challenge, of course, there is with millions of species alive on the planet, we haven't been physically able to go around and try to cross all these various species. So there's 300,000 species of beetles. No one has been able to go around and try to individually cross all 300,000 species to see who can breed with whom can't. So that's why there's all these different species definitions. So what I said in the book was the way you evaluate my hypothesis was to get the full accounting of the morphological, genetic, and reproductive separation in two candidate species species populations at two different time points. That's a mouthful of a word, but mouthful of a sentence. But basically, what I'm proposing is what you need to do is get everything that defines a species at time point 1, and pick whatever you want. How it looks, its genetics, who it can reproduce with, whatever. Just whatever defines a species, get it at time point 1, get it at time point 2. And if there's no change This would be evidence against the number I just proposed. If there is a change, that might be an argument for the formation of new species. We'll evaluate that more in a moment. For our purposes, though, and thinking back to the big picture, reasons to reevaluate Darwin, the modern debate is different than the debate of 1859. Creation science has changed. It's not the view that Darwin tried to argue against in 1859. Modern Earth creationists, like Linnaeus, who preceded Darwin, actually are okay with many new species forming within families. And what's changed within even the last 40 years is creationists have shifted from perhaps being largely anti-evolutionary, that was just an initial strategy from the 1970s. Now the creation model has a full-fledged, fully orbed, testable, predictive element to it. So it meets the standards that were required of science that have been raised within the last 40 years. That's the second reason I think it's time to reconsider Darwin, because the opposition to evolution has changed. Third reason to revisit Darwin. Those testable predictions are successful. Now I've put two terms here, retrodictions and predictions. Let me explain them. So a prediction is, I say something now, and then in the future, we'll evaluate whether or not it's true. A retrodiction is, I take data that's available to us now, and I sort of look back in time and say, which of these two ideas makes better sense of what we have currently? So I'll illustrate that more in a moment. Don't be distracted by the technical terms. But my claim here is that in both of these categories, looking forward and looking backwards, these predictions have been successful. So I'm going to give you a genetic example and then a species example. And I mentioned again, the first reason we need to reevaluate the debate is because genetics is the only direct scientific record of a species' ancestry. So now that we have genetic data, what does it say? And we're going to limit our focus in genetics to a subset of the data. If you were to take a skin cell from me and look at the genetics, it doesn't really matter because my skin cells don't get passed on to the next generation. If you want to know what matters from generation to generation, you have to look in the cells that are actually responsible for reproduction, sperm and egg. So in humans, sperm and egg would look something like this, sperm much smaller than egg. If you were to slice that egg cell in half, and look at it under microscopes, this is a diagram, but it would look something like this, very complex structure. Don't let the complexity intimidate you. All we care about for our purposes is those compartments in the cell that have genetic material. And there's two of them. The purple compartment, the nucleus, and the orange compartment, the mitochondria. Typically we think of the nucleus when people discuss DNA because it contains the vast majority of our DNA. If you think of DNA like letters, or the technical term is base pairs, there's billions of them in the nucleus. That's where you find the information that encodes hair color, eye color, height, so forth. The mitochondria, those orange compartments, are the energy factors of your cells. So if you're digesting some of the food you've eaten today and your heart's beating, it's because your mitochondria is processing what you've given your body, and that's the reason you're still breathing, because of mitochondria. They also have a little bit of DNA. It's a small fraction compared to the nucleus, only 16,569. But that's what I want to focus on for our discussion. It's smaller, it's simpler, the math is simpler. I have a whole chapter dedicated in the book to the nuclear DNA, but for sake of time, I'm just going to focus on this one compartment that's simpler. And it also has a unique mode of inheritance, which I'll describe now. So that's the egg cell. What about the sperm? If we blow it up so we can see it better, sperm also has nuclear DNA, mitochondrial DNA. And when sperm meets egg and penetrates, part of the sperm cell penetrates. Only the nucleus, the mitochondria, don't make it in. That means, practically then, your nuclear DNA is the product of both parents. So unsurprisingly, then, you have people comment, you have your dad's nose, your mother's mouth, your grandpa's ears, these sorts of things, product of both lineages. But the mitochondrial DNA comes only from mom because of this fact of cell biology, by and large, almost exclusively from mom. And if we think of this in a family tree form, again, the nucleus in purple here is from both parents, the mitochondrial DNA is in orange only from mom. Let's add one more detail to our discussion. From 1996 to 2016, there were about 20 studies that tried to measure the rate at which mitochondrial DNA changes from generation to generation. So mom in her egg cells copies that mitochondrial DNA, passes on, and occasionally mistakes happen. And the way you measure the rate at which mistakes happen is you get mom, daughter, grandmother, granddaughter. You get a whole bunch of these pairs, and you get the DNA from each generation, from grandma, from daughter, granddaughter, from mom, from daughter, and so forth. And you look for differences. And the results of these 20 or so studies arrived at a general rule. These were all done in non-African peoples, by the way. We can talk about that a little bit later. But the average rate at which mitochondrial DNA changes is one letter of our DNA, one base pair of mitochondrial DNA changes every 5 to 10 generations. So I have my mother's mitochondrial DNA, my children have my wife's. If you wanted to see a difference in my mitochondrial DNA lineage, you have to go back to my mother, to my grandmother, great-grandmother, back several generations to actually see a difference on average. Well, this rate then we can use to make some retrodictions. We have mitochondrial DNA sequences from tens of thousands of people representing very diverse ethnic groups, all the continents around the globe, which we find people. So we can now run these numbers through an equation to see if we can make sense of what we see in our globe today. So I'm going to run 3 scenarios for you, 2 evolutionary scenarios and 1 creationist scenario. Let's begin with the evolutionary one. Modern evolutionary biology would say that our closest living relative is the chimpanzee species, and that the human lineage and the chimpanzee lineage split from a common ancestor anywhere from 6 to 13 million years ago, depending on which paper you consult. The evolutionary literature has a formula for predicting then, retrodicting, I'm going to call it predicting, the number of differences that should exist today. So basically, if you have an ancestral population that leads on one hand to chimpanzees, one to humans, there's going to be this lineage of females that over 6 to 13 million years eventually gives rise to chimpanzees through reproduction. And you have another lineage that over 6 to 13 million years gives rise to humans. And every 5 generations, we're going to assume that a mutation happens, a change happens. So we can use a very simple equation. I'm a biologist, I don't like math, so all the math in this talk is going to be simple. 1 mutation every 5 to 10 generations times 6 to 13 million years, and then times 2 for technical reasons I won't get into. That should then, in a sense, retrodict the number of differences that should exist between chimpanzees and humans today. And intuitively, 6 to 13 million years is a long time, so you'd expect a lot of differences to arise. We have to do one little mathematical manipulation. The number I just gave you here is in units of changes, mutations per generation. But we want to multiply by units of years, not generations. So we have to convert that, knowing something about how old the females were when they gave birth. Well, we don't have that data. But what we do know is today, humans, for example, some women will give birth in their teens, some will give birth in their 40s. So what we'll do is we'll just calculate for that whole range, and we'll get a range of predictions. This is the result of running those numbers. Let's see if you can see my pointer right here. Yeah. So this black line represents the range of predictions given all those factors I just mentioned. So the bottom of this line represents a calculation in which we assume that all females in these lineages were age 50 when they gave birth over the last 6 million years. And over that time scale, you would get 28,000 changes and mistakes. The top of this black line represents a calculation if all females in these 2 lineages were age 15 when they gave birth. And then in between the two, the top of this bar represents sort of the average around, assuming the women were age 25, this right here. Now, someone might say, wow, the top of this line is close to 350,000 changes, but you just said there's only 16, 17,000 letters in total in mitochondrial DNA. What does that mean? What it means is you would have mutated every single 1, 2, 3, 4, 5, 6, all the way down to 16,569, and you would have done it again, and then again, and then again. That's what this number represents, mutating that almost to oblivion. The point that I want you to see, though, is that this minimum estimate is still much higher than the actual mitochondrial DNA differences between humans and chimpanzees, which are less than 1,500. Now, we'll talk about the role of natural selection here potentially in a moment. That's the first calculation, the first scenario I want to run. Second scenario is thinking about the origin of just modern humans within an evolutionary time frame. Current literature says that modern humans evolved first in Africa about 200,000 years ago, and that about 50,000 to 100,000 years ago, the non-African peoples split off. We're going to use a similar equation. The technical term is a coalescence equation, but it's basically the same form. You have a set of a population here, and the females over 200,000 years, every 5 to 10 generations, have a mistake. And again, we're going to have to calculate for women who give birth in their teens and women who give birth when age 50, and that's what this graph here represents. Once again, you can see the bottom of this prediction. If I can find my pointer, there it is. The bottom of this black line represents a calculation assuming all women were aged 50 when they gave birth. And the top of this line, come back pointer, there we go. The top of this line represents the calculation of all women were aged 15. So the bottom is close to 500 differences, the top is close to 2,500. The average then, if women were around age 25, is about 1,500. Once again, this is higher than the actual number of mitochondrial DNA differences among African people groups. So we have the DNA sequences from all sorts of ethno-linguistic groups in Africa. We have the actual mitochondrial DNA sequences from the ethno-linguistic groups of Europeans and Asians and Native Americans and so forth, and the average there is 40. So once again, this is an over-prediction. So let's stop here and think about ways these numbers might be brought into agreement. And here's a very plausible way these numbers might be brought into agreement. I just mentioned that mitochondrial DNA are like the energy factories for your cell, and the DNA in mitochondria encode some of the components that are used by your cells to break down the food you give it and use it for energy. And so it's natural to presume then that at some point, if you start messing up that DNA, you're going to interfere with energy generation, and if your heart is relying on the energy of mitochondria, eventually that's going to be lethal. So if there are some babies conceived in the womb that get a mutation that affects their mitochondrial DNA, it's lethal, they're not going to come to term. So it's reasonable to say, well, at some point, some of these mutations are not going to be seen because those babies aren't born, and at some point this process of calculating and summing these mutations is going to have to stop and reach a plateau. It's a very plausible explanation. There's others in the evolutionary literature. My challenge, though, to my evolutionary colleagues is this: whatever explanation they come up with to reconcile these numbers, what predictions does it make? So this is a retrodiction. We're using existing data, looking back, and say what makes the best sense. Now let's look to the future. That's the standard of science. What predictions does it make? I mentioned earlier that the rate of one mutation every 5 to 10 generations has been measured only in non-African people groups. So Nigerians, Kenyans, Khoisan people, the Click-speaking peoples of Southwest Africa, Namibia, no one knows the rate at which they mutate their DNA. That's something we can go out and explore and test in the future. So my challenge to my evolutionary colleagues is whatever number they come up with, whatever formula they come up with, does it make a prediction? Give me a number that we can now in the future go out and test. That's the standard of science. So those were two scenarios. Now the third scenario, the young earth creationist scenario. Young earth creationists would say that God created Adam and Eve 6,000 years ago. Adam has mitochondrial DNA, Eve does too, but men don't pass it on, so there's a mitochondrial Eve who passes it on down to all the way down through the generations of the flood. Noah, his wife, his sons, and their wives. We inherit our mitochondrial DNA from those 3 wives of Noah's sons, those lineages. Shortly thereafter, there's the Tower of Babel. The ethno-linguistic groups split and form. All that history contains 6,000 years. So how many differences should there be? One mutation every 5 to 10 generations over 6,000 years. And again, we'll calculate for a range of ages for women giving birth. That's what this graph represents. The bottom of this black line represents if all women were aged 50 when they gave birth over the last 6,000 years. The top of this line represents the prediction if all women were aged 15 when they gave birth. And notice that this range, the dotted lines here, capture the average of 80 mitochondrial DNA differences in Africans and the 40 DNA differences in non-Africans. Now, why is there more in Africans? I've put in print, there's at least 2 potential explanations. One of them deals with cultural differences. If you look at United Nations data from the 1970s, they were tracking marriage ages. And among African nations, 1/3 of African women in the 1970s were married by ages 15 to 19. And non-African nations, the women in non-African nations, only about 11%. So 33 divided by 11, it's about a 3-fold difference. That's a 2-fold difference. So if these marriage practices, cultural differences, were true historically, this might explain the difference. A second explanation that's testable, just that's one thing we can explore. Another testable explanation is, for whatever reason, African women might mutate their mitochondrial DNA at a slightly faster rate. And I've put these predictions now in print. I've predicted in my book a rate, and the prediction I made is 1 mutation every 2.5 to 8 generations or so. That's what I predict the rate of mutation will be in these non-African people, in these African people groups. So that's, I think, an illustration of the strength of the young Earth creation model. Not only is it making sense of the data that we have and looking sort of backwards in time, we can now look forward in the future, and there's ways to advance the science. That's actually only one way to advance the science. There's a second way. There's a project I'm working on right now. I hope to publish the results later this year. This diagram right here, complicated, I'll explain it very quickly. You're looking right here at a family tree type arrangement of 300 to 400 individuals. You can kind of see these lines, there's tips. Every line with a tip represents one person. I've actually got a high resolution image of this online. You can zoom in and see exactly, okay, this person is from Jordan, from Ethiopia, from Namibia, from Britain, from Germany, so forth. And the length of each line represents the number of DNA differences. So you'll notice that there's some, if I can find my pointer again here. There we go. There's some very long lines here, some shorter lines here. These long lines are the sub-Saharan African people groups, those that give rise to the 80 mitochondrial DNA differences, and these groups over here are the non-African peoples. Now, I said that all of this represents the mitochondrial DNA differences, this family tree, and I'm also claiming that all of these differences can be explained in 6,000 years. Well, in 6,000 years, the last 4,000 years, we've had the history of civilization. The Roman Empire, the Greek Empire, Genghis Khan's empire, colonialism, this transatlantic slave trade. So if all of this is a record of the last few thousand years, you should see this stamp of the history of civilization within these groups. And here's a potential illustration. For reasons I don't have time to get into, I'm going to say that from this point right here up to these tips over here, that I'm claiming that within the young Earth creationist framework that represents about 4,500 years. So about halfway up, you see a bunch of people splitting off. So I'm saying that, and then within the young Earth framework, about 2,000-ish years ago, 2,100 years ago, there was a bunch of people splitting off. Now I know exactly what countries these people come from, and I can show you here on the map where they come from. Basically North Africa, the Mediterranean, the Middle East. Well, what's happening about 2,000 years ago in that part of the world? Roman Empire. That's the type of data we should be able to see if what I'm saying is true. And the first proof of principle that I'm trying to demonstrate this with is a more recent historical event, the forcible movement of anywhere from 10 to 40, actually like 12 million African individuals from Africa to the New World. So public sources estimate that anywhere from 10 to 40% of the African continent's population was moved by the transatlantic slave trade. So we have people of African descent in the New World. We have many people obviously still in Africa. There are we have the mitochondrial DNA from African Americans. We have mitochondrial DNA from indigenous Africans. We can compare that DNA, and we should be able to see the stamp of that forcible migration. There should be some differences between these two groups in a manner that reflects 4,500-year time scale. And the reason we can do this sort of calculation is because we have historical records that are very precise in exactly who was moved, how many people were moved from Africa to the New World, which year. You can see it peaking in the 1700s. That's what this horizontal axis represents, calendar years. Slave trade starts about the 1500s, peaks in the 1700s, and then legislation finally is introduced against the slave trade. It drops off. So that's a testable prediction I put in the book, and I hope to have the results of that prediction published later this year. There are other predictions. I'm using humans as an example because they're what everyone is interested in. That's what I'm interested in. There's these same sort of retrodictions and predictions hold true for invertebrate and fungal species. I don't have time to get into those there in the book. So that's one category of successful retrodictions and predictions. There's a second category that represents actually a bona fide prediction. I published my book in fall of last year. There was a paper published in January of this year that is directly related to the numbers, the 2.4 new bird species per year that I just told you. So January was future when I published my book in October. And this particular measurement of the rate of speciation comes from Exhibit A for evolution. Darwin's finches show up in textbooks as premier example of natural selection in action? Well, there's now 18 recognized species of Darwin's finches. This is based on a paper that was published in Nature in 2015. What's been added to this story in Darwin's finches is a remarkable result, and this story begins in 1981. I'm going to show you a better image of these two finch individuals, or these two species, in a moment. This is a family tree. Circles represent females, squares represent males. So here's these lines here connecting them represent then their offspring in the second generation, third generation, so forth. 1981, a male of the large cactus finch, and you'll get a better image here in a moment, flew over to an island of the Galapagos where the intense study of evolution and natural selection was being done on the medium ground finch. So here you've got two separate species. The male comes over, mates with a number of females. Most of their offspring get absorbed into the resident population. But there's one lineage that by generation 3 here, there's close relative mating that results in offspring that stay reproductively isolated. They're building nests on the same island as that parent medium ground finch, but they're not interbreeding, reproductively isolated. That's the first thing that was observed. Second thing that was observed, and you'll see an image of this better in a moment, this lineage that's reproductively isolated is visibly distinct. They have a different beak size. Thirdly, the people doing the study, Peter and Rosemary Grant, British couple based in Princeton, New Jersey, they were getting the tissue samples. They have the genetic data from every individual in this pedigree, and this lineage is genetically distinct. So basically every criteria that defines this species was measured in this study, and the conclusion from Peter and Rosemary Grant's team in their January 2018 paper was rapid hybrid speciation in Darwin's finches, Science magazine, basically the Nature journal for the United States. Rapid hybrid speciation in Darwin's finches. So here's a better illustration. You can see here, here's a member of the medium ground finch population, so aptly named medium because its beak size isn't the biggest. Here's the large cactus finch male. There was a mating, and this big bird lineage, because it has a beak size slightly bigger than the medium ground finch, that lineage was born, new species. So what is this recorded event? What implications does this have for the predictions I made in October? Well, let's run the numbers. 11,000 bird species alive today. We ran that math, 2.4 new bird species per year. We compared it to the evolutionary prediction. But with 18 species of Darwin's finches, so they were brought to the attention of the scientific community in 1837 as a result of Darwin's voyage on the Beagle around the globe, hence Darwin's finches, that's their name. They were labeled new species by John Gould, 1837, that's 181 years ago. One new species of Darwin's finches has been discovered in 181 years. Well, 1 divided by nearly 200 gives you a number far less than 1. So, at first pass, this would seem to refute the number I just wrote. But think about this. The number I gave you was a global rate. 2.4 new bird species per year is a prediction that somewhere among the 11,000 species that fly around this planet, 2 to 3 of them are going to form a new species. And in this particular study, they've only been watching 18 of these 11,000 species, so we have to apply a correction factor to extrapolate it and compare it to a global rate. So the math you need to do is 1 divided by 181 divided by 18 times 11,000. That's the correction factor. So let's put these numbers in a row. The evolutionary prediction of 1 new species every 3,200 years is a rate of 0.0003 new bird species per year, globally, very slow. The creationist prediction is 2.4 new bird species per year. And if you do the math for Darwin's finches, extrapolate it to a global rate. 1 new species divided by 181 years, divided by 18 times 11,000 gives you a result of 3.4 new bird species per year. That's pretty close. In a sense, it's like me predicting that the Champions League football is going to still be played in the year 2050. And then me saying today, I'm going to predict for you the 2 teams that are going to play in the final. And furthermore, I'm going to predict for you the final score. And then we live our lives, we get to the year 2050. And yes, lo and behold, the exact teams that I predicted are playing in the final. And the final score is correct, except that I'm 1 goal off. You'd say that was a pretty good prediction. That's basically what this represents. So 2 illustrations that the advances of creation science, the modern debate in which we now find ourselves, are leading to successful retrodictions of predictions in multiple fields of biology. Fourth reason I think it's time to reconsider Darwin. Since October, there have been a few attempts to formal critiques of what I've published. One of them is from an evolutionary biology graduate student. He's anonymous, but he blogs at EvoGrad. So if you just Google EvoGrad, you'll find it. So in a week or two following the release of my book, it was like October 9th when the book was released, and he began blogging about the book, reviewing it October 14th. Within about 9 days, he banged off about 5 articles, reviewing chapters 1 through 6. Now, my book is laid out in 3 parts. Chapter 1 is just a teaser. Chapter 2, which represents part 1, is just a history of modern genetics. This reviewer this part. Part 2 gets into a little bit more of the controversial stuff. That's chapters 4 through 6. Chapter 4 basically just retells Darwin's argument and concludes species have migrated. And the reviewer again said, well, sadly, there's not much to object to here. Chapter 5 is where we get into more controversial stuff, but historically debated stuff. What about fossils? What about anatomical comparisons? These sorts of things that have been debated for 150 years. So we had things to say there. But it's really not much in the way of something new. Chapter 6 is where you get into new data, where you don't have cut and paste answers. And I'd say he basically misrepresented that chapter. But what I find fascinating, chapters 7 through 10, part 3 of the book, where we really get into the genetic data and what it says, he hasn't said anything on since that time. So I'm curious as to why. Now, Ancestors in Genesis is not an organization where we try to fit facts to conclusions. We have a peer-reviewed journal in which we actually publish critical reviews. Stefan Frello is a PhD botanist in Denmark. He wrote up a formal critique of my book. We published that on our webpage. You can find my response to that on our webpage as well. And if you read both, which I encourage you to do, what you'll find, I think, is that he avoided the 3 main points of my book and misrepresented the science where it got the strongest. We're now on round 2 of that, so he's got a response, and I'll have a response to his response, and we'll see how long this continues. But I'm encouraged to see people responding to this. And if you have a critical write-up, I invite you to submit this to the journal. We'll publish it, and I'll have a response. When the book came out, we sent the book to leading evolutionists. Jerry Coyne got a copy. In fact, I emailed Jerry Coyne personally, and he was very polite and professional and responded very quickly. I said, obviously we're on different sides of the aisle here, but I would be grateful if you could give me your critical, harshest feedback. And he said, well, your organization fits facts to conclusions. That's repulsive to me as a scientist. No, thank you. I said, I respect your decision. I think he'd have a different view if he read my book, but he has a copy. P.Z. Myers, who sent a copy, Richard Dawkins, who sent a copy. I haven't seen them respond yet. I will be doing a debate with that gentleman I mentioned earlier, Herman Mays, in September 5th of this year. We don't have a time yet, so I can't tell you if it's during the sleeping hours of UK or wake hours. It's the Non Sequitur show. It's moderated by 2 atheists, but what we're going to be doing here is a little bit unique. It's not a opening statement, response, that sort of cross-examination. He'll be going live on air, chapter by chapter, reviewing my book. So if anyone wants to say the book's not peer-reviewed, it will be peer-reviewed live by an evolutionary biologist, September 5. But to date, I can't find any published work that finds big errors in my work. That may change by September 5th, but we'll see. Fifth reason to revisit Darwin is that the means to refute my claims are readily available. So the book is titled Replacing Darwin, a present tense verb, because this is a work in progress, and I'm inviting people. Go ahead. This book makes predictions. Let's do the experiments to find ways to falsify it, or to see if it survives. I've already mentioned some of the ways to falsify my work. Rates of speciation, real numbers have been published. We talked about that. Mitochondrial mutation rates, I've given predictions for humans, for African people groups, for invertebrates and fungi. I talked about the transatlantic slave trade. We didn't have time to talk about nuclear DNA, but I make predictions about nuclear DNA function in the book. I'll skip point 4 because it gets technical, but there's an additional prediction there. I mentioned the prediction about the timestamp of civilization. That's ongoing. I think it'll be hopefully published at the end of this year. The point here is there are ways to test what I've just said. So, 5 reasons to revisit Darwin. Again, you can't replace Darwin in 60 minutes. That's why I wrote a book. But here are reasons I think it's time to give this a second look. Darwin took a massive scientific risk trying to answer a fundamentally genetic question long before anyone had genetic data with which to directly interrogate it. Number two, the view he overturned in 1859 is not the view that exists today. The contemporary creation science view has a different view on speciation. It's very different in its practice of science. It makes sensible predictions, and that I argue are actually successful retrodictions and predictions. That's big news if that's true. I have yet to see someone find an error, and again, I'm inviting feedback, and I've put in my book ways to do that. So you can submit. This is a free online journal, no subscription. There's no page fees, that sort of thing. I've also got a Facebook page, sort of taking a page from Darwin. When he wrote his book on the origin of species, he knew he was challenging a consensus, and he gave 4 reasons why he thought it was time to change it. He gave 4 answers to people who would say, well, no one agrees with you, Darwin, why should we? And he said, well, look, one of the reasons he gave is it takes a long time for people to change their view. People haven't changed. I don't expect it to be any different. I don't expect to change people's views overnight. And I've set up this Facebook page. The name of it is the same as my book, Replacing Darwin, for the purpose of engaging and interacting. I've had some folks come along, and there was one student in the United States who was engaging. I don't agree with you. I gave this lecture in Oxford Monday night. One of the first people came down to me was, hey, I read your book. I'm not a Christian, not a creationist. I'd love to dialogue. I said, fantastic. Let's dialogue about this on Facebook or email, whatever he prefers. So be aware of that. Point people to that if they're engaging it. You can get our book tonight at the back table. We actually have a special. We've got 2 books. The Replacing Darwin book that I wrote, again, for people who disagree with me. I'm not assuming anything about biblical knowledge. I'm not assuming anything about the Bible. It's 10 chapters of science. But it's on special tonight. That book, which normally runs for £15, and this book, Searching for Adam, also for £15. You can both of them for £20. This book is really written for pastors. Many of my evolutionary proponents, there's a group of theistic evolutionists who say, yes, I'm a Christian, but I think God used evolution. And one of their recent arguments is that modern genetic data refutes the idea that we come from Adam and Eve. It can't be 2 people, must be a population. There's big, significant theological implications of that. And this book is a multi-authored work, written again for pastors, not for nerds, to be only read by 0.2% of the population, but as a help. Each one is a standalone chapter. For example, what does the Old Testament say about Adam? What does the New Testament say about Adam? I've got a chapter I co-authored with Jeff Tompkins on what does genetics say about the origin of Adam and Eve. So you can get those, again, special deal here tonight, 2 books for £20. If you know someone you want to recommend the book to, they don't want to buy it from Answers in Genesis, you can get it on the UK Amazon site, both in print form and Kindle version. You can get the ebook form from our website as well, Answers in Genesis. Again, this is the book. One of the things I want to mention about this book, I had a pastor read through it because I wanted this book to accomplish 2 things. I wanted it to be accessible and defensible. So I had the pastor read through it and say, look, I'm not writing for my PhD peers, I'm writing for students. I want you to read through it. And he helped me with it and say, put in a glossary, explain this concept, that sort of thing. So I've designed the text to be accessible, but also defensible. So again, thinking of students, what are students going to do? They're going to read it. They might say, interesting, I haven't heard of this before. Take it to class. Professor, what do you say? And my evolutionary colleagues are going to say, well, here's the problem. So I put the technical notes, 30 pages of endnotes, to make it defensible. That's where you'll find the technical detail, which I tried to leave out of the main text because that makes it boring and leaves me only with 2% of the population. So I'm going to stop here. We're running late on time. I went late on this lecture, and I realized the evening is late, so we might have time for 1 or 2 questions. I'll be down front and happy to answer them until it's time to leave. But so if you have any questions, now's the time to do so. If you need to get up and leave because the hour is already late, please feel free to do so. But 1 or 2 questions quickly, publicly, and then we can go to...
I'd like to talk afterwards as well, but really quickly, I just was really surprised how dishonest the talk was, because I felt like you were really misrepresenting the scientific method by saying that it only looks at falsifiability, and I thought it was really telling that, there's 5 points that the judge was making reference to in what would constitute the scientific method, but yet you only focused on one and then sort of represented that as being what the evolutionist argument was, and then attacked that, and I thought that was a bit dishonest, from in my opinion. I'd love to talk to you more about it afterwards if you've got time, but from my point of view, I was a bit disappointed. I'm sorry.
So what would you say the elements of science were that I missed?
It's not just one thing, it's a method. So in addition to having falsifiability, you would have obviously predictions, you'd have the peer review process, you'd have quality of data, and so on and so forth. But it's not as though it's just one thing, and it's not to say that necessarily creation could be wrong. I just think that the argument that you're presenting isn't particularly fair, because I don't think it's representative. What I think you've done, just in what you presented, is say, this is one element that I'm going to attack, and I'm going to focus on this one element, but I feel it's like not really representative of the argument that's being made. That was all. Obviously, there's other sides to it that I didn't see. Totally happy to talk to you about it, but that's just how it came across.
I actually agree with you that there's many more elements to the scientific process. And what I tried to do was focus on the strongest ones that have arisen in the past 40 years. For example, give us a testable prediction. You can listen to those debates between Herman Mayes and the creationists, between Aaron Ra, the atheist, and the creationists. That was their main challenge to him. And I actually think creation science, the modern creationist science position, meets all of these criteria in terms of peer review and making statements about the natural world that we can test and predict and so forth. So yes, it's much more, but for sake of time, I focus on the one that I thought was the strongest one, because that's the one that's come up so frequently, and that I think many, even my contemporaries, may not recognize. So I grew up in an environment under Morris and Gish, in a Christian community where the focus was largely on what's the problems with evolution. And it's taken some time for the creation community to say, you know what, actually, what our opponents are asking for is this list of scientific characteristics. And that's been my goal in developing this model, is not just that falsifiable element, but in having it peer-reviewed, which I've solicited all along the way, and as you can see, and continuing to solicit with this debate coming in the fall and saying, go through it chapter by chapter. So,
sorry if I represented, everything you're saying in the book. It was just how this particular talk came across. That was.
Yeah. So yes, I agree, and again, can't go through all 5 points for sake of time that were in the judge's decision, but agree that there's multiple points to it. But I also think then creation science meets these 5 points. Which, if it does, is in itself, I think, a significant shift from years prior. And if it's successful, then that's another element, too. That should be part of the discussion. So, thank you.
Thank you.
One other question. We'll have to quit for sake of time, but I'll be back down front here again. If not, then I'll dismiss you. Again, we have the book table in the back. Paul and I will be down front here if you have more questions. You have ways to contact and